students, today we are going to start with chapter 5, the fundamental unit of life. We have already discussed this topic in class 8, cell, the functional unit of life. So, you are all familiar with the term cell. We had studied about the different components of a cell, the cell organelles, the differences between unicellular and multicellular organisms, eukaryotes and prokaryotes, and also the differences between plant cell and animal cell. Here in this chapter, we will be once again redoing it and also going one step further to go into the details of the various cell organelles, their functions, and also the different processes that happen in the cell. So let us begin. The cell was discovered by Robert Hooke in 1665 while he was observing a slice of cork. Cork is the bark of a tree called Cocos suber. He examined this slice of cork through a primitive microscope and found honeycomb-like structures. And these honeycomb-like structures resembled like tiny rooms. Therefore, he named it as cell. Let us refer to activity 5.1. We need to take an onion bulb, cut it into half, and take one of the halves and separate the fleshy scale leaves. Then we need to take a thin membrane out of the concave side of one of the fleshy leaves and immediately transfer it to a petri dish containing water. We need to stain it with a few drops of safranin. Then, using a brush, we have to transfer this peel to the center of a clean glass slide. Then, pour a few drops of glycerin over it. Place a cover slip and view it. We will see that it has similar structures. All the cells are similar, and these tiny compartments which we see are known as cells. We can also see the cell wall and the nucleus is darkly stained, while the cytoplasm and the vacuole is lightly stained. What can we conclude from this? All organisms are made of cells, and whatever be the size of the organism, but the cells are the same. Whatever size the onion bulb we take, but when we observe it under the microscope, it is of the same size and it is similar. Now, let us see the contributions of various scientists. As I told you, the cell was discovered by Robert Hooke in 1665 under a primitive microscope. Leeuwenhoek used a more progressive microscope to find free cells in pond water. Robert Brown discovered the nucleus, Purkinje coined the term cytoplasm. Sorry, not cytoplasm, protoplasm. Sheldon and Schwann proposed the cell theory. Now, what is the cell theory? Cell is the basic unit of life and all organisms are made of cells. It was Virchow who modified the cell theory and he added one more statement to it. That is, cells are formed from pre-existing cells. In 1940 was the big discovery of electron microscope, which allowed us to study the various cell organelles in detail. Now, organisms can be unicellular or multicellular. What do you mean by unicellular or single-celled organisms? They have only a single cell and all the work is done by that single cell. So there is no division of labor. The examples are amoeba, paramecium, euglena, all are protozoans, and chlamydomonas, which is a unicellular algae. While multicellular organisms are made of many cells and there is division of labor, that means a group of cells are present, different types of cells are present, different groups of cells combine to form tissues and tissues into organs and organ systems. Now cells, they are of different shapes and sizes. The shape and size of a cell determines the function of a cell. Let us see a few examples. The spindle-shaped involuntary muscles, cells, help in involuntary actions such as breathing, movement of the intestine, etc. The RBC is spherical and contains the red coloring pigment hemoglobin. Neuron or the nerve cell is branched and elongated. It sends and receives messages. Ovum is spherical. It is the female gamete. While sperm is tiny and it has three parts: head, middle piece and tail. It is the male gamete. The bone cell is also known as osteocyte. It resembles somewhat like an ice crystal and it gives support. And lastly, the fat cell, which is otherwise known as adipocyte. So we have seen various examples of different types of cells found in humans. The shapes are different, the sizes are different, and thus their function also differs. Cells are made up of different types of cell organelles. These cell organelles help the cell to live and perform various activities. The cell organelles of different cells all are the same. The function of all cell organelles, whatever be the cell, is the same. A cell has three basic components, that is the plasma membrane, the cytoplasm and the nucleus. Now let us focus on the plasma membrane. The plasma membrane is the outermost covering of a cell. It is selectively permeable or it is semi-permeable. What does this mean? It means that it allows only certain molecules to move in and out of the cell. We need to discuss the term diffusion. It means the movement of molecules. It can be gas molecules or liquid molecules from a region of higher concentration to a region of lower concentration. The movement of carbon dioxide and taking in of oxygen during respiration is an example for diffusion. That means carbon dioxide is given out and oxygen is taken in. During photosynthesis, carbon dioxide is taken in and oxygen is given out. Both these examples are examples of diffusion. Then what is osmosis? Osmosis is the movement of molecules from a region of higher concentration to a region of lower concentration but across a semi-permeable membrane. Now, let us discuss three types of solutions. That is hypotonic solution, isotonic solution and hypertonic solution. Now, what do you mean by hypotonic solution? If we place a cell in a dilute solution, we see that the movement of molecules, especially water molecules, is from outside to inside the cell. And as a result, the cell swells up. The size becomes larger. The solution has more amount of solvent and less amount of solute. Therefore, the molecules, that is the water molecules, move from outside to inside. This is known as end osmosis. And this type of solution is known as hypotonic solution. Next, let us move on to isotonic solution. In such a solution, if we place a cell, there is no change in the shape of the cell because the concentration of the water inside and outside is the same. So the net result is that there is no movement across the cell, inside to outside. This is because the amount of water that goes out and comes in is the same. So the net result nullifies. Such a solution where the concentration of the solution inside and outside is the same. That means the amount of solvent and solute outside and inside the cell is the same. Lastly, hypertonic solution. In a hypertonic solution, if we place the cell in a concentrated solution, that means solvent is less and solute is more, then the movement of molecules will be from inside the cell to outside. In such a case, the cell will shrink or the size will become smaller. And this type of movement is known as exosmosis, and such a solution is known as hypertonic solution. Now, to understand osmosis further, let us discuss activity 5.4. We need to take an egg and put it in dilute hydrochloric acid so that the shell dissolves. So the egg is only left with the membrane. We take this carefully and put it in pure water. We will see that the egg swells. This is because the movement of water molecules is from outside to inside. That is, endosmosis takes place. We take another de-shelled egg and place it in concentrated solution of sugar or salt. We see that the egg shrinks. This is because the movement is from inside to outside. Next, let us also discuss activity 5.5, where we are supposed to take a few dry resins or apricots and place it in water. When we do that, we see that the resins and the apricots, after a time of 1 hour or something, you can see that it swells or the size increases here again, because the movement of water molecules is from outside to inside. Again, if we place these dry resins and apricots in concentrated solution of sugar or salt, we can see that it shrinks and becomes smaller because the movement of molecules is from inside to outside. That means exosmosis happens. Now, unicellular aquatic organisms and plant cells take in water through osmosis. Not only this, the roots of plants also absorb water by osmosis. Let us see how diffusion is important to the cell. It helps to take in gases and water, and also it helps to absorb nutrients from the environment. Different molecules move in and out of the cell using energy. This type of transport is also known as active transport. Now, plasma membrane is flexible and is made of complex organic molecules known as lipids and proteins. This plasma membrane, because it is highly flexible, it helps to engulf food. This we can see especially in the case of amoeba. Amoeba is a unicellular organism which does not have a definite shape. That means the plasma membrane is highly flexible. Therefore, it can put finger-like projections called pseudopodia and engulf the prey when it comes in contact with the prey and incorporate it into the body and forms the food vacuole. So this process by which the amoeba catches its prey using the flexibility of the plasma membrane is known as endocytosis. Thus, we have come to the end of today's session. We will be continuing with the components of the cell and discussing in detail the different cell organelles in the next session. Till then, thank you.